Assalamualaikum warahmatullahi wabarakatuh. And good evening to everyone. So today we are going to present our SCL infographic of the topic of active transport. So my group members consist of myself, Arisa Balkis, and we have Anis Rabia, Nurin Atira, Aisha Shahira, Alif Makira, and also Serafia. So let's start our presentation with the subtopic of carrier protein that will be presented by Alif Makira.
In order to understand active transport, so it is necessary to understand first about the carrier protein. Carrier protein is the protein that was used during active transport by moving the solid against concentration gradient by the help of ATP. The mechanism of carrier protein is similar to the enzyme substrate reaction, and each of the carrier protein has their own binding site for their solute. So there are three types of carrier-mediated transport, which are uniport, symport, and antiport. Uniport transport a single solid from one side of the membrane to the other, and it is classified as primary active transport if it uses ATP. For example, glucose carrier uniport that transport glucose from extracellular fluid into the cytosol. Next is the symport. Its transport of one solute depends on the simultaneous or sequential transfer of the second solute in same direction. For example, Na plus ion and glucose symport. Next is the antiport. Antiport is transport of one solute depends on the simultaneous or sequential transfer of a second solute in the opposite direction. Difference between symport and antiport is the direction it involved. If symport is the same direction and antiport is the opposite direction. Example for the antiport is Na plus and K plus antiport. Next, there's a figure that shows a model of how a conformational change in carrier protein that could mediate the positive transport of a solid. The carrier protein shown can exist in two conformational states. In state A, the binding site for solids are exposed on the inside on the outside of the lipid bilayer, whereas in state B, the same sites are exposed on the other side of the bilayer. So the transition between the two states can occur randomly, and it is completely reversible and does not depend on whether the solid binding site is occupied. Therefore, if the solid concentration is higher on the outside of the bilayer, more solid binds to the carrier protein in the A conformation than in the B conformation. And there is a net transport of solid down its concentration gradient. So that's all. Thank you.
Thank you, Alia Fakira, for the carrier protein sharing. So I would like to explain my part, which is active transport. Okay. Active transport, almost all plasma membrane have an electrical potential difference across them, with the inside usually negative with respect to the medium. This potential difference favors the entry of positively charged ion into the cell, but opposes the entry of negatively charged ion. Okay, as we know at this picture, the Na plus concentration is higher outside the axon than inside. So as for potassium concentration is higher inside the axon than outside. So cells also require transport protein that will actively pump certain solute uphill against their electrochemical gradient. This is known as active transport and always mediated by carrier protein. Uphill meaning is the concentration was from high to low. The solute is moving from low to high concentration. Okay, active transport can be driven by the energy stored in ion gradients or by ATP hydrolysis. And may be a uniporter or antiporter. Okay. Active transport usually use ATP hydrolysis directly or indirectly. to move molecules across the membrane up or against the concentration. So as Alia mentioned just now, we have uniporter, symporter, and antiporter. For uniporter, the solute was moving straight. But for symporter, the solute and ion, couple ion, will move also straight, but two of them will move together straight. And we for antiporter, the solute and ion will not the same way, like against each other to move. Okay. The gradients also maintain many life processes, right? Like negative and positive. So it processes the life processes to move. So this picture show that antipart and sympart move, how they move. The antipart will move against each other, but sympart will move in one direction. So I think that's all from me. Thank you.
So I'm Nurati Rabinti Zulkifli. We present about sodium potassium ATPase pump. So in animal cell have a lower sodium ion and higher potassium than ion than environment. So in this system, the solute and the Na plus will bind to the different side on a carrier protein because the Na plus tend to move into the cell. So for animal cell, this effect is counteracted by opposite Na plus and Cl negative in the extracellular fluid. So the Na and Ka ATPase maintain the osmotic balance by pumping out the Na plus, while the Cl negative is kept out by the membrane potential. So how it works is, so the sodium potassium pump is worked by bind 3 Na plus and a molecule of ATP. Then the splitting of ATP provide the energy to change the shape of the channel. Then the sodium ion are drive through the channel. Then the sodium ion are released to the outside of the membrane, and the new shape of the channel allow two of the potassium ion to bind. So lastly, release of the phosphate allow the channel to revert to its original form. Then releasing the potassium ion on the inside of the membrane. Okay, that's all.
So I will continue with my part, which is a type of ATP powered pumps. So the type of ATP powered pump we have is, the first one is that can hydrolysis of ATP for the P class, B class, and ABC superfamily. The second one is that the pump that synthesize the ATP, which is F class pump. The P class pump, P present for phosphorylation. For V, the V class pump present in backward. The ABC superfamily, it present the ATP binding cassette. For the synthesis of ATP, the F class pumps, it's present in mitochondria, chloroplast, and bacteria, which is they will produce the ATP for the use of the cell. So the next part is the Ca2 plus pumps. Ca2 plus pump is the P class pump. So some of the Ca2 plus pump also membrane bound ATPase. It is important to the eukaryotic cells to maintain very low Ca2 plus concentration in their cytosol compared to the much higher extracellular concentration of Ca2 plus in the cytosol. So the best understood Ca2 plus pump is in the sarcoplasmic reticulum, which is SR. SR can store the Ca2 plus. When an action potential depolarized, the muscle will release SR will release Ca2 plus into the cytosol so that it can stimulating the muscle to contract. In the muscle contraction, Ca2 plus will function as for the need of myosin to bind to actin in the sliding filament model. So for the ATP, it supplies the energy for the muscle to contract. So that's all for me. I pass to the next presenter.
For the next part is proton pumps. Okay, so the plasma membrane bacteria, the inner membrane of mitochondria and thylakoid membrane of chloroplast contain enzyme that's similar to the ATP to transport ATPase, but it works as reversely. So instead of ATP hydrolysis, hydrogen gradient was generated during the electron transport system of oxidative phosphorylation. So in aerobic bacteria and mitochondria. And then ATP synthase is actually the transport ATPase. So ATP, the enzyme that synthesizes ATP, the ATP synthase, will hydrolyze the ATP and the pump proton across the membrane, or it can synthesize ATP when the proton flows through the enzyme in the reverse direction. Okay, and then next we have the ABC transporter superfamily. So this is the last type of carrier protein that were found and they were characterized in bacteria. So the PM of all bacteria carrier protein use proton gradients to pump a variety of nutrients into the cell. So some also have transport ATPase that uses the energy from sugar, peptide, and amino acid. Okay, and then as for the picture shown, it is shown that it is the ABC transporter superfamily that is located in the bacteria plasma membrane, and it is called as the ABC transporter superfamily because each member of the protein contain a highly conserved ATP binding cassette. Okay, and then lastly, the ABC transporters have been described with like more than one specific to a particular substrate, which means that one ABC transporter can only bind to a specific substrate. Okay, I think that's all from me, and next I will pass to the last presenter.
So the last part of our presentation is the secondary active transport. So it's another form of active transport. The transporter protein couples the movement of an ion as Na plus or H plus down its electrochemical gradient and another molecule or ion against its concentration or electrochemical gradient. This type of transport is called secondary active transport because active transport means that it uses ATP, and secondary means that it's the second transport who uses ATP. So energy is stored in the electrochemical gradient during the first movement, and it's then used by the second one, which means the first ion transported is called the driving ion. It drives the uphill movement of the driving ion or molecule, which is the molecule or ion transported against its concentration or electrochemical gradient. In addition, there are two variations of secondary active transport. Firstly, the co-transport. The driving ion and the driving molecule or ion pass the membrane in the same direction. They both enter or leave the cell. Secondly, the exchange transport. The driving ion and the driving molecule or ion pass the membrane in opposite direction. In this case, one enters and the other leaves the cell. That's all for our subjects. Thank you for listening.